So in the last lesson, we saw that broadly we can think about experimental design as having two different categories. So treatment design and randomization design. And in our study of ANOVA, we've discussed some basic features of treatment design, like how many treatments are there, how many levels does each treatment have, etc. And in this lesson, we're going to start off in our study of randomization designs. So in particular, we'll describe the simplest experimental design, which is called the completely randomized design, or CRD. And we'll consider what assumptions we need to hold in order for a CRD to be reasonable. And we'll later on supplement this video with an analysis of some real data using a CRD. So let's first say something about randomization. So in a randomized experiment, experimental units are assigned to an experimental group, for example, treatments and controls, by a chance process. And the chance process could be assigning a Bernoulli random number to each unit, or it could be some other chance process. And just as a note, being assigned a control means something like being assigned the status quo or baseline. For example, in medical trials, patients who receive a placebo are considered a control group. And in social media, say in a social media advertising experiment, when a company wishes to test the effectiveness of the current advertisement against the same advertisement with a new color scheme, the current advertisement with the current color scheme is the control. And there are several variations on randomized experimental designs, and the most basic experimental design is the completely randomized design, which we'll introduce now. In a completely randomized design, or CRD, treatment levels or combinations of treatment levels are assigned to experimental units so that each experimental unit had the same chance of receiving any treatment. So that's sort of what characterizes a completely randomized design, the fact that the random assignment, every unit has the same chance of getting the treatment level. So as an example of a completely randomized design, consider the espresso data set that we began analyzing at the beginning of this unit. And recall that the goal of this experiment was to evaluate and compare the differences in terms of quality between espresso coffee made using 3 different extraction procedures. So one was this bar machine method, the second was the hyper espresso method, and the third was the iEspresso system. And quality, remember, in this experiment was measured in terms of the foam index. And foam, according to the authors of the study, is a distinctive feature of espresso coffee, as it is absent from other coffee brews and is required for consumer acceptance. So this experiment can be thought of as a completely randomized design. The treatment is the brewing technique, and that was assigned randomly to experimental units, which you can think about as measured volume of espresso. So let's think about some advantages and disadvantages of a completely randomized design. So the CRD is a great design in many contexts. It's flexible in that, at least in theory, the number of treatments and replications are only limited by practical considerations, such as the number of experimental units a company or research lab can afford to study. So it might be the case that the coffee company who's studying those different machine brewing techniques can only afford to, dedicate so many dollars to this experiment, and so they might be limited by that. Completely randomized designs are also easily analyzed by ANOVA techniques. And in cases where we have one treatment at potentially many levels, a CRD can be analyzed using one-way ANOVA, as was the espresso experiment. And with one treatment of relatively small number of levels, the degrees of freedom for the residual sum of squares, which remember is the number of experimental units minus the number of treatments, will be large. And I'm getting that degrees of freedom for the residual sum of squares from the regression context that we've looked at all semester, right? And I'm just thinking about it in the ANOVA context with the dummy variable model that we've looked at. So the CRD also has some drawbacks. It's inappropriate if we have reason to believe that experimental units are not homogeneous. For example, if we had reason to believe that different volumes of espresso were different in some way that affects foam index, then a completely randomized design would not be appropriate. Another disadvantage of a completely randomized design is that although it's easy to apply mathematically, it's often not appropriate mathematically because homogeneous experimental units are difficult to find. So often there are things that are different about units that we can't quite detect. And if we apply a completely randomized design, say using one-way ANOVA, we might be making an assumption that they're homogeneous when in fact they're not. Finally, although theoretically the CRD is flexible with respect to the number of treatments, in practice it is best suited for a relatively low number of treatments because with each additional treatment we lose a degree of freedom. So will students soon study experimental designs that allow us to overcome these disadvantages? For example, we'll look at block designs that allow us to overcome the problem of heterogeneity. But in the next lesson, we'll analyze some data coming from a CRD.